Hi, everybody. Thank you for joining today. We'll give another minute for people to roll into the meeting, and then we'll get started. Welcome, everybody. Yes, please also, before we get started, please use this time to introduce yourselves in the chat. We have people coming from India, China, the APAC region, so it's a good chance to network and get to know each other. And then in a few seconds, we'll get started. Okay, great. I think we can get started. Yes, hello again, and welcome to the first session of Alt Protein Dialogues, a new webinar series designed to connect and showcase alternative proteins innovators across Asia Pacific, India, China, Australia, and New Zealand. Alt Protein Dialogues is a collaborative initiative co-organized by GFI APAC, GFI India, and GFIC. Our goal is to foster regional partnerships that drive globally impactful innovation in the alternative protein sector. My name is Arun Naidu, and I'm the Senior SciTech Ecosystem Specialist at GFI APAC. I'm thrilled to be your host for today's session as we kick off this series with a topic that's essential for advancing alternative protein research: the GFI Research Grant Program request for proposals. This two-part webinar series will provide a detailed overview of the 2025 Research Grant Program, including its focus area and application process. So part one, which is today's session, will be an application process overview. We'll cover the fundamentals of the RGP, the key research themes for this year, and guidance on the application timeline and submission process. Part two, on April 16th, will feature past GFI grantees who will share insights into their experiences with the program and practical advice for applicants and how to apply. Whether you're from academia, industry, or government, this session will equip you with the knowledge to craft a strong application and take advantage of the funding opportunities available through GFI. The session today will follow go through the following structure. The first 40 minutes will be introducing the RFP and grant topics. It'll be a recorded session from GFI US, where our colleagues there will introduce the GFI Research Grant Program, its objectives, and the topics for the 2025 grant call. We'll then have a 10 to 15-minute Q&A session. We will open the floor for questions from the audience. Feel free to submit your questions in the Q&A box throughout the webinar, and we will address as many as we can. To address the questions, we'll have Dr. Wasamon Nutakun from GFI, who's GFI APAC SciTech Team Manager. And then we'll also have Chandana Tekate, who's GFI India SciTech Team Manager. Before we dive in, a quick housekeeping note. You'll see the chat and Q&A button at the bottom of your screen. Please use the Q&A function to submit questions for our speakers, and then feel free to introduce yourself in the chat. If you see a question that resonates with you, upvote it so we can prioritize the most relevant ones. This webinar will be recorded, and we will share a recording along with additional resources after the session. With that, let's begin. I'll now start by playing the recorded session from our GFI colleagues introducing the GFI Research Grant Program and this year's funding priorities.
Hello, and thank you for joining us today to learn more about GFI's Research Grant Program. We're very excited to share more information about our 2025 request for proposals. By joining this webinar, you're taking an excellent first step down the pathway to success. In terms of our agenda for today, we'll start by providing an overview of GFI's Research Grant Program before diving into the specifics of our two funding priority areas. After that, we'll share more information on how to apply before opening it up for questions. First, some housekeeping information. Please be aware that all participant lines have been muted. If you have any questions during our presentation today, please submit them using the question and answer box displayed at the bottom of your screen. To help out our moderators, when submitting your questions, please follow the formatting examples displayed on the screen. If you prefer to ask your question via email, then please direct it to us at rfp@gfi.org. Additionally, it's worth mentioning that this webinar content has been pre-recorded. If you're watching this recording during one of our information sessions, Then we'll have a live question and answer session following this presentation. Depending on the specific session you're attending, the team members that will address your questions during our Q&A may be different than those featured within this presentation. These dedicated global members have graciously volunteered their time to enable additional information sessions, and we're incredibly grateful for their support. I'd like to quickly introduce myself. My name is Elisha, and I'm proud to serve as GFI's new Research Grant Program Manager. I'll be introducing you to GFI and our Research Grant Program today. The Good Food Institute is a 501c3 nonprofit developing the roadmap for a sustainable, secure, and just protein supply. Powered by philanthropy, GFI is an international network of organizations advancing alternative proteins as an essential solution needed to meet the world's climate, global health, food security, and biodiversity goals. We have earned GuideStar's 2024 Platinum Seal of Transparency, which is an honor obtained by less than 1% of nonprofits. Our organization employs over 200 team members across the globe with 3 key programmatic departments. Our science and technology team, which includes today's presenters, serves to advance foundational open access research in alternative proteins and create a thriving research and training ecosystem. Our corporate engagement team focuses on driving investment, accelerating innovation, and scaling the supply chain. Through their collaborations, these efforts are executed faster than market forces alone would allow. Lastly, our policy team advocates for fair policy and public research funding for alternative proteins. And together, we play a unique role and uplift stakeholders across the alternative protein landscape. Through our research grant program, we support essential early to mid-stage research proposals designed to solve many of the most timely and pertinent challenges facing alternative protein technologies. We seek to create open access tools and methods for the development of appetizing, affordable, and widely available alternative protein products. Our research grant program is dedicated to achieving 3 core objectives: addressing technical bottlenecks, unlocking and catalyzing further research funding, and fostering collaborations to attract new talent to the field. To achieve these goals, we annually release requests for proposals inviting open access research focused on enhancing organoleptic properties, reducing costs, and scaling up alternative proteins. GFI established our research grant program in 2018, an effort made possible by the incredible generosity of GFI's philanthropic supporters. Since our launch, we have provided yearly opportunities for researchers to apply for rapidly deployed funding, which have gone on to support 129 projects across 25 countries by providing over $24 million in research grant funding. As previously mentioned, we're firm in our dedication to advance foundational open access research for wide-scale use. Thus, we formulated our grant agreement in such a way to enable this effort. Though GFI encourages information sharing, we do not prohibit the generation of intellectual property or its protection. Our intellectual property terms vary based on organization type. We provide an additional lay summary of our intellectual property terms within the RFP's FAQ section. We highly encourage all prospective applicants to review our standard grant agreement, which is linked within the RFP, as they prepare to submit their proposals. This year, we're offering up to $3.5 million for our 2025 RFP cycle to support innovative open access research across 2 priority areas. Priority A is addressing functionality from fermentation ingredients for plant-based meats. For this topic, we are asking for proposals to identify and characterize fermentation-derived ingredients that enhance alternative protein-based meat products to increase their adoption through improved taste, lowered cost, and nutritional benefits. Priority B is pathways to propel cell line development. This topic includes 3 potential pathways: fish, and terrestrial. We are asking for proposals to advance cell line development to bolster innovation and strengthen open access research tools for the cultivated meat ecosystem. Now I'll hand it over to Adam to share a little bit more about Priority A. Adam?
Thank you, and good day, everybody. Thank you for your interest in our research grants program. And today I'm going to talk you through our 2025 funding Priority A, functionality from fermentation, ingredients for plant-based meats. My name is Adam Lehman, and I'm a principal scientist in fermentation here at GFI. So we know that in the future world of alternative protein formulations, that ingredients will come from a variety of sources. In our research, we often split alternative proteins into three main pillars: plant-based proteins, fermentation-derived proteins, and cultivated meat. However, we know that many formulations will and do include ingredients from other pillars to achieve functionality, nutrition, and organoleptic properties. For example, we know that fermentation-derived ingredients are used as growth factors for cultivated meat. And we know that fermentation-derived enzymes can be used in plant protein processing to improve that protein functionality. However, we believe that most future and next generation end products will contain ingredients from another pillar. These can include ingredients such as flavorings, an additional source of protein for nutrition or functionality, and fats. And we believe that fermentation can provide these. Plant-based meats have attractive nutritional profiles and range from partially to mostly mimicking the organoleptic properties of animal-based meats. Taste, texture, mouthfeel, cooking transition behavior, such as a change in color from red to a more cooked brown, aroma, and other characteristics have greatly improved over the past decade. But how can we improve on any one of these specific organoleptic challenges further? We believe that fermentation-derived ingredients have reached a maturity and a diversity where they should allow for increased functionality for plant-based meats. In a timely publication, Nectar has released this month the Taste of the Industry 2025 report. And I encourage you to read this report. And on page 17, you can see that there's a quote that asks for increased upstream innovation in ingredients or processing to close the gap on functionality and organoleptics, increasing the sensory appeal of plant-based meats in many different types of meat formulations, such as nuggets, burgers, sausages, bacon, etc. And we know that fermentation has long filled roles in increasing the functionality for foods. Taking an example of heme binding proteins that bind the iron binding molecule heme, we can use recombinant protein platforms, precision fermentation, to produce this key molecule that can improve ingredient functionality and plant-based meats like plant-based burgers and formulations like that. We also know that we can use metabolic engineering to produce volatile compounds that are identical to those given off by cooking meat. So you can imagine recapitulating the aromas and textures and even sounds of meat cooking with these novel compounds. We can also improve the taste and texture of plant-based meats through new fats and new fat encapsulation methods, which allow for plant-based meats to hold on to the fats that are included in those formulations and deliver that fat taste, texture, and aroma that come along with retaining those fats. We can also use microbes to biofortify particular formulations by keeping higher levels of vitamins or minerals in those ingredients and then passing them on to that final end product. An example of where precision fermentation has increased product formulation is with the fermentation-derived soy LGBA protein. Precision fermentation-derived soy LGBA was approved by the FDA in 2019 and is an important ingredient for plant-based ground meat from Impossible Foods. This is a molecule that you can see here, the 3D structure. It's an ingredient that adds iron content by binding and delivering heme and iron to plant-based food formulations. It provides a meatier taste, and it also undergoes an oxidation-mediated color change when cooked, going from a red to a cooked brown color. And we believe that other proteins, flavor molecules, and fatty acids may similarly improve plant-based formulations in other ways. Biomass-derived protein, such as mycoprotein, provides texture and protein concentration to alternative meat and dairy. Although in this particular funding priority A, we are not funding alternative dairy at this time. However, mycoprotein's particular attributes make it an attractive choice for meats. It has long fiber structure that's made by a filamentous fungi, and it often has high protein content, sometimes around 30% dry weight for its ingredient formulation. While alternative dairy products have been commercialized with a mixture of plant-based and fermentation-derived ingredients, Fewer efforts have been made to use mycoprotein or other single-cell proteins as a plant-based meat ingredient. Fundamental research could provide insight into how single-cell proteins can improve plant-based meats. And here from this review by Thomas Linder, What's Next for Mycoprotein, we can see commercial examples of where mycoprotein has been used in formulations. However, only in the plant-based and fermentation-derived yogurt shown in panel D do we see a mixture of plant-based and fermentation-derived ingredients that significantly increase the protein content, but also the organoleptic appeal of that alternative yogurt. So what are we looking for our funding priority A, functionality for fermentation? First, we are looking for proposals to articulate the challenge for plant-based meats that they seek to solve or to improve, and then to describe how the inclusion of a fermentation-derived ingredient addresses this need. GFI encourages innovations related to biomass and or precision fermentation ingredients that improve plant-based alternative meat formulations. Just going to walk through two examples. Biomass fermentation, where whole cell mass is inactivated, dewatered, and texturized to provide a high protein content cell biomass, can be added to alternative protein meat products as a high protein ingredient, could also act as a binding ingredient, or could be optimized to drive flavors such as umami or provide meat-like textures. Within an alternative protein meat, biomass-derived protein could complement plant-based ingredients by diversifying the amino acid content of end products and providing essentially amino acids that can be rare in some plant-based protein ingredients. To date, precision fermentation-derived ingredients such as globin proteins have been formulated successfully to improve the iron binding and organoleptics of all protein meats. There's an opportunity for other precision fermentation-derived proteins or other ingredients to specifically increase the appeal and enable the production of AP meats, such as alternatives to egg white protein binding ingredients that are sometimes available in alternative proteins formulations. These precision fermentation-derived ingredients could enable improved binding, water and fat retention, texture, aroma, taste, or nutritional value, such as mineral delivery like iron, zinc, magnesium, etc., for meat alternatives. So what are we looking for our proposals? Again, proposals should address end product formulation, characterization of those end products or the ingredient to be used in the end product, and research plans for experimental assessment of fermentation-derived ingredients used in plant-based meat formulations. Examples include, but are not limited to, nutritional content, digestibility, functional properties, and food science applications such as emulsification, gelation, water and oil holding capacity, biochemical, biophysical properties like the color, the salt content, or other molecular values, sensory appeal, and food safety. Successful proposals must articulate which ingredient that falls within the above guidelines will be the focus of the project, with a specific target meat alternative and product formulation and assessment. We're looking for the food formulation challenge to be articulated and for the investigations to relate to the functionality and or organoleptics that you seek to improve. Then we would like to know the approaches that you will specifically investigate on how the particular ingredient will address that food formulation challenge that you described. And then we would like a clear description of the ingredient's potential as an available ingredient to address the identified organoleptic and functional challenge. These ingredients used should be the ingredients that are possible to produce, that could be potentially produced at scale within the next few years that would make this an appropriate solution for the plant-based meat space. Next, we would like to describe the types of proposals that we are not looking for our funding priority A for 2025. First, we will not consider proposals that focus on cultivated meat ingredients that either are going to be derived from or used in animal cell cultures to produce cultivated meat products. We are not looking for ingredients aimed at animal feed applications. We are not looking for proposals focused on ingredients used as high protein flours for other food applications beyond meat alternatives. This can include alternative proteins that could be formulated into pastas, bakery, protein supplements, or confections. We are not looking for formulations focused directly on alternative dairy, meat, or cheese products. We are not seeking ingredients that rely on the transformation or conversion of animal-derived products by microbial fermentation. Examples of this could include fermentations driven by animal-derived lactose or whey protein. And we're not looking for proposals using animal-derived ingredients or upstream feedstocks and inputs. We're also not looking for proposals focused solely on the production of microbially derived fats. And lastly, we are not seeking proposals focused on R&D, scale-up, or production of the ingredient. For topic A, GFI seeks to advance R&D on improving the organoleptic properties of meat alternatives through formulation with enabling fermentation ingredients. We're looking for proposals centered on food science, specifically the formulation and characterization of these ingredients in meat alternative applications. While ingredient production and bioprocessing are critical R&D areas, this RFP focuses on end product development to enhance taste, texture, and other sensory qualities. Projects may include ingredient production and modifications that improve the ingredient, but the primary emphasis must be on integrating and characterizing the ingredient into meat alternative formulations and applications. Next, we'd like to share some resources that you can use in advancing your proposal. So please check out the Nectar Taste of the Industry report that I described. Also, there are several publications here which give examples of a review from GFI on precision fermentation food ingredients, but also other examples of where precision fermentation and biomass fermentation ingredients could provide functionality to plant-based foods. And lastly, we'd just like to share some examples of past work that has been funded by GFI that fills some highly similar roles. So you can see more at gfi.org/grants. And next, I'd like to turn it over to my colleagues in Cultivated Meat to describe funding priority B. Thank you.
Thanks, Adam. Hi, everyone. My name is Claire. I'm one of the cultivated meat scientists at GFI, and I will be presenting funding priority B along with my colleague Manasa from our APAC affiliate office. All right, so our focus for this particular priority is cell line development for cultivated meat for 3 different species groups, so crustaceans, fish, and terrestrial animals. We have some slightly different considerations for the 3 of those, which we'll get into. All right, so the main challenge that we're addressing here is the lack of available cell lines with desirable characteristics that we would want for research and commercial purposes for cultivated meat. And this is really a persistent bottleneck that's holding us back, not just from commercializing cultivated meat products, but also from doing a lot of the more sort of basic level research that needs to happen in academia to build up the foundation for this whole industry to exist. And so depending on the species you're working with, the level of challenge and what we think is likely to be feasible in 2 years will be a little bit different. And so to reflect that, we've customized the core objectives for each of these 3 pathways. And also, depending on the species focus of your work, we may also encourage or require the inclusion of a secondary objective on top of the core objectives. And so Manas is going to explain a little bit more about what those secondary objectives might be later on. But the basic idea is that we would like you to take things a step beyond just developing or optimizing a cell line and use that cell line to enable further experiments that will solve another downstream problem in cultivated meat or seafood. So pathway 1, we've divided this topic into 3 separate pathways. So pick the one you're focusing on and kind of focus on the criteria for those because they will be a little bit different. Pathway 1 focuses on developing and optimizing myoblast or myosatellite cells from crustaceans. And so because there's been less foundational research done on crustacean cell culture up to this point, the core objectives for this pathway are the simplest of the 3. So the first core objective for this pathway is simply to develop a continuous myogenic line and to demonstrate its differentiation to muscle cells. The second core objective focuses on media development. And so since we realized that achieving fully serum-free media may be a little bit overambitious for a 2-year project on top of the cell line development angle, we're very much open to approaches to reducing serum in the media as opposed to completely eliminating it as a first step, especially for crustacean projects. And through the third core objective, we're seeking to provide an opportunity for researchers to help improve the field's understanding of really what cell line stability means. So currently there's a little bit of a lack of international consensus and guidance on how cell line stability may be defined, its applicability and suitability within the context of food safety, and the type of data or best practices that are sufficient to demonstrate it. So we encourage you to sort of put on your evidence-based thinking hats to develop an experimental strategy to define and show that your cell line is genotypically and phenotypically stable. And finally, the fourth objective focuses on continuing efforts to improve the accessibility of cell lines. So what this means is you are required to bank or deposit your cell line in a repository of your choice, as well as add it as an entry into GFI's cell line database. Secondary objectives for this pathway are completely optional, since, as I mentioned, we realize you may have your work cut out for you with the core objectives. So you're welcome to include a secondary objective if you think you can manage it, but the main thing we're looking for here is projects that have a really solid chance of succeeding on the core objectives. So my advice would be to please be mindful of that when deciding whether to include a secondary objective or not. Pathway 2 focuses on developing and optimizing either myoblasts or myosatellite cells or adipogenic progenitors from fish. So the first core objective for this pathway focuses on the development and optimization of the cell line and media. And so as with the first pathway that we just talked about, we're looking for a continuous line that can differentiate into the relevant cell types. So in this case, that would mean muscle or fat, and also at least some first steps toward reducing the serum content needed in the media. It doesn't necessarily have to get all the way to serum free for this one. One new thing we're asking for in this pathway is adaptation of the cell line to grow in either suspension or 3D culture systems, as well as efforts to bring down the doubling time, ideally before 24 hours if you can manage it, but we realize that that might be asking a lot. As with pathway 1, the last two objectives focus on defining and assessing genotypic and phenotypic stability, and on ensuring that the cell line that's developed through this project is broadly accessible to the cultivated seafood community. So depositing it in a repository. For pathway 2, we strongly encourage you to include a secondary objective if feasible. However, we recognize that this could be a little ambitious depending on the other details of your project. So this is still considered optional. And as with pathway 1, if you're not sure if you'll be able to pull off a secondary objective, I would advise erring on the side of having a really solid plan for the core objectives. Try not to spread yourself too thin by picking an overly ambitious secondary objective. Consider kind of the trade-offs that might come with that. And with that, I will turn it over to my colleague Manasa to talk about pathway 3.
Thank you, Claire. Hi, everyone. Pathway 3 focuses on developing and optimizing continuous cell lines from terrestrial species across several different cell types. The core objectives laid out for this pathway build on those that you saw in the previous slides for pathways 1 and 2. The first core objective is to ensure that the developed cell line possesses characteristics that are deemed important for scaling cultivated meat production. These characteristics include growth in serum-free media, growth in suspension or other 3D culture formats with a short doubling time, ideally less than or equal to 24 hours, differentiation down myogenic and adipogenic lineages, and growth in feeder-free conditions for embryonic stem cells and induced pluripotent stem cell lines. The second and third core objectives are consistent with the other two pathways. You are required to develop an experimental strategy to define and show that your cell line is genotypically and phenotypically stable, and also bank your cell line in a repository of your choice, together with adding it as an entry onto GFI's cell line database. In contrast to pathways 1 and 2, addressing a secondary objective in addition to the core objectives listed here is essential if you pick pathway 3. I'd like to now go into a little bit more detail about the secondary objectives. The purpose of our requirement to address a complementary secondary objective is to provide an avenue to push your research a step beyond just creating another cell line. We are keen to drive the so what of your research. So after you develop your line, how can you actually use it to advance the field? What downstream research question or bottleneck can your cell line help answer or alleviate? With this in mind, we have provided a list of suggested secondary objectives in the RFP But here are just a few examples that you can consider. One, identifying an approach to reduce the production or accumulation of toxic waste metabolites in suspension or 3D culture systems without hampering cell line stability. This could be through cell engineering or media recycling, for example. Two, comparing different published protocols or improving methods to enhance proliferation or differentiation down myogenic or adipogenic lineages. Three, collecting dry mass, biomass composition, stoichiometric, and kinetics data from relevant species and cell types grown in different bioreactor environments to inform life cycle assessment and techno-economic assessment modeling. You may propose an objective that is outside of the list provided and consider a problem statement that you feel you are well equipped to tackle based on your expertise and capabilities. However, please ensure that the secondary objectives selected is clearly justified and relevant. It's feasible within the project timeframe and doesn't detract you from fulfilling any of the core objectives listed in your particular pathway. As a reminder, it's optional to address a secondary objective for pathway 1. We strongly encourage it for pathway 2, and it's essential for pathway 3. So ultimately, what are we looking for? Applicants are urged to carefully consider the current state of the science, the commercial relevance, and open access merits of their research. So we're looking for proposals that clearly highlight the pathway, species, and cell types on which you want to focus your research on and why. If your proposal spans more than one pathway, please highlight one of them as your primary focus. Select a complementary secondary objective and justify the rationale as is appropriate for your chosen pathway. Describe the proposed experimental strategies clearly and share supporting preliminary data if you have any. It's not a must, but we would love to see any preliminary supporting information that you may have to achieve the core and secondary objectives. Include a plan to document details related to the provenance of the cell line to the extent that is possible for your chosen species and your cell sourcing method. Some examples of details to include comprise animal health particulars, vaccination history, source tissue type, anatomical location, evidence of veterinarian assessment, and so on. Please include a plan to bank the cell line and showcase a willingness to share it with the cultivated meat and seafood research community. Consider the commercial applicability of your research by highlighting a strategy on how you think the cell line developed will help close the gap for cultivated meat or seafood to market readiness. Lastly, you can outline a plan to document compliance with halal or kosher standards. This is completely optional and is only encouraged if it is feasible and appropriate to do, given your chosen species, cell sourcing method, and experimental approach. This really is a good to have, not compulsory. So please note that proposals will not be disqualified or disadvantaged if this is absent. So what are the sorts of proposals that we would not be considering? The focus is on developing a cell line from a terrestrial fish or crustacean species that is not food relevant, and if the choice of species and cell type is not adequately justified. So to clarify this, cell lines from insects and organisms without a history of large-scale industrial farming or fishing for human consumption, or focus on cell types not relevant to meat or seafood, this could include, say, mammary cells, would be out of scope. Second, any of the core objectives stated for a selected pathway are excluded. A relevant secondary objective is not selected or justified for pathway 3, or proposed experimental strategies are not described adequately. 3, attempt to replace serum with insect hydrolysates or other animal-derived materials. However, proposals aiming to replace serum with albumin as an intermediate step, but not the final step, may be considered. Proposals lack a plan to A, document details related to the provenance of the cell line, and B, bank and share the cell lines developed from this project with the broader cultivated meat and seafood research community. We also would not want to see a situation where the commercial applicability of the research outputs is ignored. So we highly encourage applicants to think about this carefully in your proposals. And finally, if the proposal refuses to acknowledge a willingness to add the cell lines developed from your project to GFI's cell line database, we would not be able to consider that. In the interest of time, I won't go through these one by one, but if you are keen to do some background reading to learn more about this funding topic and the context behind why we've chosen to focus on it this year and how we've thought through it, please check out the list of resources we've provided in the RFP, which is the same as what you see on the slide. These include reports and resources developed within GFI, and as you can see on the next slide, related peer-reviewed journal articles and research projects that we have previously funded on a related topic. With that, I would like to wish our applicants all the very best in preparing your proposals. We look forward to receiving them. And with that, I would like to hand it back to Alicia. Thank you.
Thank you all for that helpful breakdown. I will now walk our audience through our application process. Before getting started with your proposal, please read our RFP linked on our Research Grant Program webpage. Next, please log into our proposal portal. If you've not applied for our grants before, then you will need to create a free user account to get started. To do so, please follow the instructions displayed on the portal homepage. The deadline to submit your application, including all proposal components, is noon Eastern Daylight Time on May 15th, 2025. We will not accept proposals after the submission deadline. It's worth noting that once you have submitted your application, our portal will allow you to access the application and make edits up until our stated deadline. Additionally, we strongly encourage you to take note of our other requirements. Projects may not exceed 24 months, and applicants must be prepared to initiate their projects within 3 to 6 months of the submission deadline. Budgets may not exceed $250,000 US or $300,000 US maximum if your proposal will include a new to the field collaboration as defined within our RFP. All proposals must be written in English and adhere to all stated page limits. I want to be clear that GFI's RFP is available to researchers worldwide, and we encourage all eligible submissions regardless of your geographical location. We recognize that this requirement means that many prospective applicants may be writing in a non-native language. Please do not let this discourage you from applying, as all proposals will be evaluated based on their scientific merit, not language proficiency. Lastly, in accordance with US law, GFI is unable to consider individuals or organizations currently sanctioned by the United States government for research grant funding. GFI verifies applicant compliance with this requirement during our review process prior to sending any award notices. All applicants are required to agree to provide supplemental information if requested to enable GFI to confirm your compliance with this requirement. The next few slides serve to document the required proposal components. All applicants will be required to provide details about their project, including organizational information, their project title, summary, abstract, key personnel, and information about proposed human subject, animal, and hazardous material use. A document detailing your research design is also required and may not exceed 6 pages. A maximum of 1 page of this document must describe your research team, summarizing their qualifications and background. The remaining 5 pages should be dedicated to describing your project aims, research design, methods, any collaborations, figures, and references. All applicants are required to submit a work plan, which may not exceed 1 page. Your work plan serves to outline your project objectives, milestones, tasks, and dependencies included in your research design, and should include general timeframes rather than specific dates. All budgets must use GFI's approved budget template, which is linked within the application, to itemize costs by budget category stated in US dollars. Applicants should request the funding required for their project success while building a conservative yet realistic budget. Applicants without collaborations are limited to 1 page in length. Applicants with collaborations may include 1 additional budget page per sub-award for any collaboration. You must also submit a separate budget justification. This document is limited to 2 pages in length and must detail and justify the resources you are requesting to support your project, including any sub-awards. If you have requested the additional $50,000 available for proposals that include new to the field collaborations, then you must also provide greater detail regarding how your collaborator meets our new to the field definition. A separate document, not to exceed 2 pages in length, is also required to describe your impact strategy. The document serves to provide a detailed description of your plans to achieve meaningful outcomes and maximize the impact of your proposed research. I encourage you to read the FAQ located within our RFP for additional information on the impact strategy document. Lastly, we provide the option to upload 1 or more letters of commitment, which are limited to 2 pages per letter. Please note that these letters are considered optional unless the principal investigator is a graduate student or a postdoctoral fellow. Eligibility for these applicants is contingent upon the inclusion of a letter of commitment to be signed by a faculty member from their higher education institution. Those letters must explicitly affirm the faculty member's commitment to act as a collaborator and advisor for the project. They must confirm the proposed research will have access to the necessary laboratory facilities. And if the applicant is a student, they must include the current aims of their dissertation and describe how the proposed research will contribute to it. When applicable, applicants should submit a letter of commitment to document any committed financial or in-kind resources for the project they will receive from either their organization or other collaborators. Those letters should define the financial or in-kind resources committed to their project and describe the collaborator's role in the project. All submitted proposals undergo a comprehensive evaluation and are evaluated using the same criteria. Our primary review criteria, the category we hold in highest regard, is originality and significance. To assess this criteria, our reviewers will assess the novelty, innovation, and transformational potential of the research, its relevance to the funding priority area, and the suitability of your proposed methodology. Moving on to our secondary review criteria, our reviewers will assess the research team based on the researcher's ability to deliver the project as proposed, including the expertise and experience of researchers and collaborators, the balance of skills within the team, and how strong, meaningful, and synergistic your collaborations are. In terms of project planning, our reviewers will look to see that you have clear, well-defined research objectives and your methods are clear, feasible, and capable of producing reliable results. Our reviewers will want to see a well-structured plan that clearly details your research stages, timeline, and any key milestones. It should also come as no surprise that our reviewers will evaluate the value of your proposed project against the financial resources you have requested from GFI. They want to see a reasonable budget for your proposed activities with rational, transparent justification. As a reminder, applicants are encouraged to submit conservative yet realistic budgets to accomplish all work as described within their proposals. Lastly, our reviewers will evaluate your project's expected impact. They will look to see how comprehensive, feasible, and creative your impact strategy is, and evaluate your work's potential for achieving a meaningful and/or broad impact on the alternative protein landscape. This concludes our presentation. We hope this information has been helpful as you consider submitting a proposal for funding consideration. By now, you may have some questions for us. If you are joining us live and have not already done so, we encourage you to submit your questions now using the question and answer box displayed at the bottom of your screen. In doing so, please label your questions as either a process question or with the specific priority area, A or B, that you are inquiring about. Additionally, I want to provide a friendly reminder that many questions are already addressed within the frequently asked questions section located at the bottom of our RFP. If you have additional questions after today's event, please read our FAQ to see if your question has been already answered. And if not, please email your question to us at rfp@gfi.org. Thank you for joining us today. We will now open the event up for a live question and answer session.
All right, thank you everybody so much for your attention. I hope you all now have a better understanding about the 2025 GFI 2025 Research Grant Program, more understanding of the research focus areas and research themes covered in this year's program, how to apply the proposal components and evaluation criteria. As mentioned during the discussion, please make sure to check out the grant program website, the FAQs page, and also if any details were missed, definitely worth considering rewatching the recording, which will be uploaded on YouTube later. And a big thanks to our global GFI team for putting together the grant program and preparing this presentation to share to all of us. And yeah, as of now, I'd like to open the floor up to Q&A. So please put your questions in the chat box. And today, as mentioned prior, we have Dr. Wasmon who is our SciTech team manager from GFI APAC, Chandana, who is the SciTech team manager from GFI India, and we have Dr. Manasa Ravi Kumar, who is the senior SciTech specialist for cultivated meat at GFI APAC. And we all just heard her speak for a little bit too. So yeah, I think we can I'll pass the mic over to Dr. Wasmon, and we can start addressing the Q&A.
Thank you, Erin. I just saw one question that comes up. A few questions come up, so please keep the Q&A coming. But the first question that comes up right now is, does confidential data can be submitted? Do you keep confidentiality of the proposals? For the proposal, yes, we are keeping your proposal confidential, so we will not be using any of the data of your proposal for any other work within GFI. However, after you are granted GFI funding, one of the requirements of our grant fund is to share your knowledge in the open access. So I think you have to evaluate whether your data, your confidential data, would that be part of your publication, open access publication, and evaluate whether that's usable data for you for this particular proposal. But within the proposal review and proposal process, everything will be kept confidential.
And
So,
oh yes,
Chandana, I think that this one I'll share for you. So the next question is it possible to have multi-country partnership? Are there any limitation for the number of partners, nationally or globally?
This is a very good question, and the reason we are doing this GFI Asia wide all protein dialogues webinar is to encourage more partnerships. So yes, you can have multi-country partnerships, and as far as I know, there are no limitations to this. So if we go to page 23 in the FAQs document, in the RFP document, you will see that the various types of collaborators you can have, whether it's from the same organization, a different organization, and there is a distinction between whether they're a service provider or a proper collaborator on your project. So if it's a proper collaborator, then they will receive a subcontract from GFI. And if it's a service contract, this may be included in the budget separately in the services category. So yes, you can have multi-country partnerships. You can have them as collaborators between countries, and you will have to specify on what type of collaboration they are. Are they a service provider, or are they a proper co-PI in the budget? So that is something you'll have to pay attention to. And yeah, if you need more details, please go to page 23 in the RFP document, and you'll see the full details.
Thank you, Chandana. So the next question I have here is from Sally Wilson. Thank you for a great webinar. I'm sure it's covering FAQ. Just curious regarding the financial contributions required from the applicants and associate industry or government partner. What type of cash or in-kind allocation be required from applicants? Sally, can you amplify your question? I'm not sure I fully understand your question.
I think Sally might be asking if there's any inputs required financially from the applicants themselves to like secure the funding allocation?
No, not necessary. However, you can, like, one of the other questions we use to get whether GFI funded can be used as part of the other funding program. So it's possible that we have a separate work package that is funded by GFI, and then you might have a different work package that's funded by other funding. That is possible, but for GFI, we hope that the GFI funded is its own separate project, but it might be under like a umbrella program, for example.
And I think the clarification point is, will GFI favor applicants with some of the projects if some of the project is co-funded also by other applicants?
No, I think we would prioritize project according to scientific integrity of the proposal. So all the proposal will be compared against each other in that way. Yep. We have a next question for Manasa. So this question is cell line. What defines a continuous cell line for this call?
Hi, yeah, that's a really good question. So we have kept it a bit generic by using that term simply because there may be different sort of thresholds or cutoffs in terms of number of passages or population doublings that are possible for a particular cell line, depending on the pathway that you choose and the species. So ideally, what we would like to see is sort of persistent growth in vitro without hitting senescence. You know, if there's a way you can showcase that you've actually maybe tested that, and an example would be looking at senescence markers and seeing that they don't change over a period of, you can select a sort of culture period of your choice. So we're also allowing freedom to the researchers to also develop how they want to define that. But for terrestrial, for example, I would recommend taking a look at what some of the cell lines already existing out there, what sort of population doublings they have, how long they've been in culture for. That some of that data is publicly available in, say, the regulatory approval documents, for example, in Upside and GoodMeat. So if you can kind of actually reference one of them as like an example and say, okay, we're going to be following these thresholds, that would be totally justifiable. So we haven't really fixed an idea saying, oh, it has to be X number of population doublings or anything. All I would just say is within what makes most sense for your cell line culture system and the species. And I would also like to just add that, for example, if you're working with an embryonic stem cell line and an induced pluripotent stem cell line, just if you have preliminary data where you've already worked with that line on something else for a certain number of days or, you know, population doublings, you can use that as evidence. So yeah, I hope that answers that question, but you can also send us, if you have more specific questions, just send us an email and we'll get back to you with more targeted answers.
Yep, and that email will be to rfp@gfi.org.
Thank you, Melissa. We have a question on the open access request, whether the open access request is for the granted proposal or the result of the research. The open access requirement from GFI grant is for the result of your research that is funded by GFI grant. And I have the next question on priority A. So does priority A also include colorant based on fermentation? If yes, can you please elaborate on this? So priority A focus on the application of fermentation product. So if you have a fermentation target, like such as the colorant that is produced via fermentation, and you can showcase its application in the plant-based meat metrics, you can be eligible for priority A, because we really want to highlight the application of plant-based meat and not in other food categories. So please read carefully in the RFP within the priority A of what is within the scope and what is not within the scope. So mainly we're looking for any fermentation ingredients that can enhance organoleptic properties of plant-based meat or alternative protein-based meat products.
Yeah, and maybe I'll just add, I can quote a little bit from the funding priority A, what the team is looking for. Proposals should address end product formulation, characterization, and research plans for experimental assessment of fermentation-derived ingredients used in plant-based meat formulations. Examples include, but are not limited to, nutritional content, digestibility, functional properties such as emulsification, gelation, water, and oil holding capacity, chemical and physical properties such as color, salt content, and then sensory appeal, as well as food safety, example being allergenicity.
So we don't have any other question in the Q&A box. Chandana, is there any last point you would like to cover before we go off?
Yeah, thanks, Mon. One question that we have noticed is often asked is about what makes one eligible as new to the field and how they can be able to win that extra $50,000. What makes them qualify? So we'd be, I know this, I'm very surprised to see this question hasn't come today because it's a very popular question every year. So we'd like to highlight on that aspect, exactly what qualifies someone as new to the field. So this is very elaborately mentioned in page 20. Yeah, sorry. New to the field is mentioned in page 23 under eligibility. So here you would qualify for the extra 50,000 only if you haven't priorly received funding for doing any alternative protein research. Your partnership, whoever is your new collaborator, would not have previously published on this or would have a different type of skill set that brings a new interdisciplinary approach to your proposal. So we really encourage that you find such collaborators who are new to the field and bring a fresh perspective to your project. Please go through the RFP in detail for what type of collaborators are required for both priority topics. It'll give you a good idea on what are the unique kinds of edges that we're looking for. I see there was, yeah. And Mon, is there anything else you think we should highlight to the participants?
I think maybe one of the aspects that might not be as familiar in our region is the impact statement. So I would highly recommend everyone to go and check out the impact statement section within the FAQ, which I believe is listed like almost at the last page of the RFP. But within the FAQ, they will have a few prompts that you can utilize to think of idea to generate your impact statement, like a very strong impact statement for a GFI proposal. Thank you, Warren.
Yeah, please make sure to check the FAQ because there's a lot of really good information on there. Yeah. I think with that, we can wrap up our Q&A. Any other points from our panelists today?
Any other point from Manasa?
No, maybe one other, just a small thought, because we've had a couple of inquiries on this. For each of the pathways for topic B, you are welcome to work with more than one cell line. So at the very least, we require one cell line, but if you would like to, if you're already somebody working on cultivated meat and seafood lines and you want to use that as sort of a comparison for comparative analyses for a new line, etc., you're more than welcome to develop those kinds of strategies. We're not averse to, you using this also as a chance to optimize something that you've already been working on. So this may also include, species comparisons if you want to compare the same cell type growing in the same conditions but from different species, for example. So you are welcome to explore the boundaries of what you can maximize this opportunity for in your proposal. We're not being super prescriptive. For us, it's just important that the core objectives and as appropriate, the secondary objectives are addressed in some shape or form. So yeah, just wanted to highlight that.
Wonderful. Yeah, thank you so much to our panelists and experts here for giving a little bit more insights, addressing the questions, and hopefully giving a better sense about how to apply for this program. And with that, I want to thank everybody here for attending part 1 of the GFI 2025 Grant Call Application Insight webinar series, and the first part, the first All Protein Dialogues webinar bringing together our GFI India, GFI APAC, and our GFIC audiences together. Huge thank to our speakers for sharing their experiences. And I do want to say that everybody here attending today, we really do believe that all of you are the ones with the technical expertise and the ability to really solve the bottlenecks in the all protein industry. So our grant program is here to enable and empower all of you to do that wonderful work. So we really encourage you all to apply. And to get more of these detailed insights, we do hope that you join us next week for our part 2, where we'll hear directly from past GFI grantees, people who have gone through the application process, been achieved the grant, and are have been working on their research projects as part of the GFI grant program. So you can ask them more questions, you can get more insights about what their experience has been as well. And yeah, many thanks to everybody involved in making the webinar happen today. And thank you again, all of you all, for attending. You'll receive a follow-up email with recording of today's sessions, relevant links, and details on how to register for part 2. Thank you all again, and we look forward to seeing you soon. Have a good rest of your day, evening, and yes, have a great rest of your week. Thanks for joining. See you.